Hello, you guys. Today we're going to be solving the November 2020 IB Biology Paper 1 paper. So let us begin by solving. So for the first question, we have the electron micrograph shows a section through a cell, and it refers to questions 1 and 2, meaning that you have to look at questions 1 and 2 when you're answering. Okay. So let us begin. So we have, what is the name of the cell component Y, right? And this is Y right here. We have Y. So we need to know what this component is, right? So option A, we're going to be using elimination for this one. Option A, it is the Golgi apparatus. No, it is not, because the Golgi apparatus is much smaller in size. Plus, the Golgi apparatus is in charge for protein, distribution and protein transportation, and it usually looks something around that shape. So no. The second one is nucleus. Yes, it could be potentially the nucleus because firstly it has a double membrane. I don't know if you can see it, but this is really double membrane and it's large in size, so it could be the nucleus. But remember, you always have to read all the options. Cytoplasm, no, it is not the cytoplasm, because cytoplasm is usually like empty spaces throughout the cell, which are in charge for the chemical reactions of life. So it's not the cytoplasm. And the vacuole, no, the vacuole is usually much smaller in size and it's just in charge for storing food particles and whatnot. So it's also not the vacuole. So our answer for this question is, in fact, B. And now for number 2, which feature of the cell in the micrograph is consistent with the endosymbiotic theory? So in this question, what are they asking us about? The endosymbiotic theory, right? Now, let's break down the endosymbiotic theory. So we have the mitochondria and we have the chloroplast. So we have these two organelles, right? So here we already have identified Y as the nucleus. So automatically we see a Y and Y. We cross them out just because the two organelles for the endosymbiotic theory are the mitochondria and chloroplast. Now, what is this symbiotic theory exactly? It's that the mitochondria and chloroplast were originally each individual prokaryotic cells, but they were engulfed by eukaryotic cells, right? So here we can tell that these two have 70S ribosomes. They divide by binary fission as well. So they have all the features of prokaryotes, right? So X, which we can deduce is the chloroplast because we can see the chlorophyll inside, and the chlorophyll usually gives off pigmentation under the electron micrographs. So now we can automatically know that this is a chloroplast organelle, and the fact that they're asking about the endosymbiotic theory also hints that it is the chloroplast organelles. So here, X has a single membrane, but this is not the point of the antibiotics theory, endosymbiotic theory, sorry. So here, X contains 70S ribosomes is our answer. So now we have question 3. Which sequence has the cells arranged according to their ability to differentiate? So first we have differentiation, very important, starting from the least able. So here they're also giving us a sequence because in IB they also test your focus. So you need to be able to actually read the question well and understand what they want. You need to know the grammar behind the question. So differentiation. So firstly, differentiation is basically how a cell can differentiate, meaning you have embryonic, which differentiate a lot. You have adult stem cells, which are limited. You have specialized cells, which cannot help really in differentiation because they're already specialized. Okay, so now we need to actually read our questions. So now we have the least ability. Keep in mind the least ability. So we need the ones that differentiate the least. So basically we need a cell that is specialized, right? So the first one we have bone marrow, neurons. So we automatically know this is wrong because neurons are actually extremely specialized cells. And then you have question, well, part B. It says neuron, bone marrow, umbilical, embryonic, which does make sense because embryonic stem cells are the ones that differentiate the most, and they're the end here. So now we know that this is a correct answer because neuron is the first one, which is correct because neurons are the ones that are specialized the most, hence they differentiate the least. The third one has neuron in the end, last one has neuron in the end, so don't waste your time on these. And question B is answer B is the right answer. So now we have question 4. So here we're talking about modes of transport, right? So we have two modes of transport. We have the second mode. I directly can see that there is a channel protein, meaning that this is going up a concentration gradient. So it's going up a concentration gradient, right? So now we directly know that this is going up a concentration gradient, so it's not osmosis, whereas this is is not facilitated by anything else, right? So we directly begin looking at our answers. So the first answer has simple diffusion and osmosis. Simple diffusion can be considered correct for the first one. However, osmosis is false because osmosis is actually going down a concentration gradient and it does not require a channel protein. So does not require channel protein. Then our second one, where we have B, active transport for the first one. No, because this cannot be active transport because there's no channel protein and it's going down the concentration gradient, so no. And B is facilitated diffusion. Maybe. Facilitated diffusion can be considered correct. C, simple diffusion, facilitated diffusion, both are correct. And as you can tell, we did them from question A's and B. So when we're eliminating, we're actually saving ourselves more time. So then you have facilitated diffusion, active transport. These are both wrong. This is not facilitated diffusion. D is not facilitated diffusion because it needs a channel protein. So moving on. So question 5 is that we have how many chromosomes are there in a cell during the anaphase of mitosis? So this is very important because mitosis is one of the cellular divisions, but we also have meiosis. So in this question, they're specifically asking us about mitosis. If the diploid number, so diploid number is when it is 2n, keep that in mind, of the cell is 20. So 2n equals 20. Once you start breaking down the question, you'll actually really understand what they want and how you'll answer it exactly. And we have chromosomes. Keep that in mind, chromosomes. Okay, so in the anaphase part, we have here. Okay, here we have the microtubules and we have the sister chromatids, which have been split into the opposite ends of the cell, right? So now each sister chromatid has become its own chromosome. So what we do is that we end up multiplying 20 by 2. And here we get 40. So our answer is C. It is 40. Okay. And then for our next question, number 6, my apologies. And we have what type of reaction. So they want the type of reaction and the products shown in the reaction. Great. So directly when we look in the reaction they gave us, we can directly see that in the products we have H2O, which is basically water. Water has been released from the reaction. Once H2O is released from the reaction, we know that this is directly a condensation reaction because water is one of the products. It's a product in condensation reactions. So here we directly look at our answers. We can directly cross out reaction hydrolysis because this is not hydrolysis, this is actually condensation. So now we have to look at the products. We have either 2 nucleotides, which is false because we literally have one molecule as our product. And then we have one dinucleotide, which is true because 2 nucleotides have formed into one dinucleotide, right? So our answer is B because we can tell that these 2 molecules became one molecule. So it's one dinucleotide. Then we have question 7. So here, this is the chart shows the range of body mass index BMI. Very important. A 20-year-old boy has a height of 120 centimeters, and always, 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 very, very important to keep in mind of the units you have, and weighs 28.8 kilograms. What weight category is he in according to his BMI? So this question is quite tricky because usually someone would think to just use your x-axis, your y-axis, and your pain point, which is usually most of the questions. But in this case, that's not the question. We have to actually calculate the BMI. So BMI is equivalent to weight. In this case, it's kilograms, so kilograms. And then we have a height squared. But keep in mind that the height is in meters, not centimeters, right? Okay, so the squared is obviously not the parent, so I'm just gonna square it here. So your weight is 28.8. We did not touch it, but okay, but the 120 turns into a 1.2, in fact. Okay, so here we have 10 to the minus 1, and then squared, so it becomes 28.8 over, or actually we'll just like get the final answer now. So our answer is, in fact, 20, right? So now we know that his BMI is, in fact, 20. But one second, guys, I'm just fixing one thing in my calculations to ensure that it's correct. Yeah, it is 20. So here we have 20 for the BMI, because we realized that our y-axis is a BMI, it's not the height. And here we have age years. So we know that the age of the boy is 9. So here we put 9, and then we just keep on going until we meet the 2 points. Okay, so this is basically it. So they meet at this point eventually. This point is in the category of overweight. So the answer is C, the boy is overweight. So now let us move on to question 8. So the genetic code is shown. In the coding gene, the DNA triplet in the transcribed strand is changed from AGG to TCG, okay? What would be the result of this change in the genome? So now we need to actually see what they really, really mean from the table that they have given us, right? So let's go. So we first see, we first see the first base, which is A in this case. Okay. Second base which is G, and third base. So we go all the way here, this area, right? Then our third base is also G, so we have ARG, arginase. And then we go to TCG. So we have to go to TCG, where we get as well A. Wait a second, guys. So we know that adenine goes with thymine, and then cytosine goes with guanine, right? So we need to make sure that this is exactly how it is. Yes. So we do know that first we also get, so I'm going to do it in another color. So A, then we go all the way to guanine, and then we go all the way to A again, so we get arginase. I think this is it, guys. Because our answer is that there would be no change to the protein. So this is our answer, because the protein would actually stay the exact same in this case. So this is why we cannot really change it. Because T is in fact A, and C is in fact G. Okay, so this is how it stays the same. And then we have for the next question, 9, the diagram represents transcription and translation. what do structures, what structures do the letters X and Y represent? So as we can see that X is actually the like it represents the mRNA because look where it's located, on which strand. And then we can see that Y is actually the anticodon because it's going towards the mRNA. So our answer in this question is B. So question 10, what is the reason for tag DNA polymerase being used in the polymerase chain PCR reaction? So basically what happens in the polymerase chain reaction, so basically what happens in the PCR is that it's the replication of DNA because they're placed in really hot temperatures. So then the DNA strands begin to unwind. So unwinding of DNA, in case you want to just review this section. So I'm trying to like break down these questions and help you guys review for them. So for this question, the first question is that why they used specifically tag DNA polymerase is exactly their question. So we need to extract their question. So they say it does not denature at high temperatures, which is true, and it ends up unwinding the DNA. It produces Okazaki fragments more rapidly. It allows translation to proceed rapidly. It works efficiently with helicase and PCR. So all of these do not specifically answer the reason for tag polymerase being used because the reason is that it does not denature in high temperature and it does unwind DNA. So the answer is in fact A. Okay, this is why, because the fact that it produces Okazaki fragments is true, but that does not mean that it's the reason they use tag polymerase, you know? So yeah. Now, the diagram shows a respirometer. So here we have these small organisms, okay, and then we have the glass beads. So in this question, what solution should move in the bottom of each tube, and in which direction will the manometer move? Okay, so keep in mind these are just a bit annoying because they start telling you left side, right side, right side. That's it. So just keep in mind, you know your right side and your left side. You need to just look at the image itself. Okay, just look at the image itself, right? So in this case, it will go down on the right side. So it's going to go up on the left side because you need to think of it this way. The fluid usually goes towards the organism, right? So it's gonna go up on the left side and it's gonna go down on the right side, right? So you need to eliminate the ones that say otherwise. So when the one that says down on the left side is wrong, so you eliminate D, or if it says up on the right side, it's wrong, so you eliminate C as well. So now you're left with two options because it is true that it goes up on the left side, it is true that it goes down on the right side, right? So now we need to know the solution placed in the bottom of each tube. We know that in this case it is alkali because when they ask about their respirometer, always know that the conditions in fact are alkali and they're not acidic in order for the organism to survive. Then for the next question, plants produce carbon dioxide in respiration and use carbon dioxide in photosynthesis. This is true. The graph shows the volume of the carbon dioxide exchanged in plants at different light intensities. So here in the y-axis, we have carbon dioxide exchange, and the x-axis, we have light intensity. Keep that in mind. So what is shown in the graph? There is no photosynthesis between W and X. Okay, so here they're trying to show us that because there is no light intensity, because it's negative, it's below zero, that photosynthesis won't take place. But that is not accurate because it might have stored light energy before and it's undergoing photosynthesis, so it's not completely accurate. And again, we have to reread the question to get the wording. Plants produce carbon dioxide in respiration and use, okay, so here they're just reminding the candidate, so they're being kind enough, the IB, to remind the candidate what's happening. The graph shows the volume of carbon dioxide exchanged in a plant at different light intensities, okay? And then there is no photosynthesis between Y and Z, which is technically not possible because it has light intensity. The light intensity is positive, okay, even though there is no carbon dioxide exchange, whereas here there is carbon dioxide exchange. So this is why this proves that this is not true because photosynthesis can still be taking place because there's the carbon dioxide exchange, whereas here they're like like arguing it because they're showing that there is no light intensity. There is a light intensity, but there's no carbon dioxide exchange. But the fact that they do have light intensity, but there's no, like, it's not increasing, just shows that there's enough light intensity. The plant does not need more. So these two are wrong. And then for answer C, sorry, there is more respiration than photosynthesis between Y and Z. So no, there is no proof for that. So there is more respiration than photosynthesis more respiration than photosynthesis between W and X. This is true because there is more carbon dioxide exchange, but there is less light intensity. So this is the answer. And this is why they showed us this on the x-axis and this on the y-axis. So this is our answer. So question 13 says, in the pedigree chart, individuals affected by genetic disease are shown as shaded in the symbol. So when they're shaded, it means that they are affected with the disease, right? And we know that squares are males and circles are females. This is something you should know while you're studying for your IB exams. What is the mode of inheritance of the genetic disease? So now we have to know if it's sex linked, or first we need to know if it's sex linked or it's autosomal. Then you need to know if it's dominant versus recessive. So these are the two categories that you should always tackle when you are solving pedigree chart questions, right? So here we can directly tell that we have one that is not affected. Okay, so guys, I'm gonna start circling the affected ones, just showing you the affected, and I'm gonna use another blue. So here we know that this is like one's affected, one's not affected, but we know that some of the children are in fact affected, right? Um, so we do know that actually it is dominant. So we have to cross out the ones that have recessive. So it's not B and it's not C, right? Now we need to figure out whether it's autosomal or whether it's sex-linked, right? And how can we figure that out? So this is actually quite easy in this case. I kind of already cracked it. So here we can see that the mother, which has the chromosomes XX, these are her sex chromosomes. Okay, has XX. So her son, and this is dominant, right? So her son has to inherit one X from her, right? So maybe he inherited the recessive one, right? So they didn't show us the disease, so this is just X recessive, I guess. Whereas the daughter, she inherited the X dominant because she gives each one an X, and she has to give this son one of her X's. But since it's dominant, one is dominant and one's recessive, right? But for the male, it's quite different. Why? Because here, for the mother, she is X, X, right? So we do know that she has recessive, recessive, right? But she has to give her son one of her X's, right? Because her son has sex chromosomes of XY, right? The Y comes from the father, okay? And the X comes from the mother, but the mother is recessive, hence she's homozygous. Hence, this is not sex linked because his XR. So he cannot be affected. So this is not a sex linked disease. And it cannot be like only a sex male. Like, for example, sometimes it's with the Y chromosome, but here females are affected with this disease. So you automatically know that it is not sex linked, it is A. So our answer is A for this question. So now for 14. So the question says the heterozygous, this is very, very important, tall heterozygous pea plant were crossed and the resulting seeds grown. Out of 300, okay, so both are heterozygous, tall heterozygous. Out of 360 plants, 270 were tall and 90 were dwarf. What describes the expected genotype resulting from the cross? So here we have, so this is T is for tall, T is for dwarf. Deal. So T, 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 T. So we have T, 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 T. So here we can automatically tell that those with these genotypes, the genotypes that contain this gene, are automatically all taught. So 75%, or 1 out of 3, or no, sorry, 3 out of 4, right? Whereas in this case, 25% are short, 1 out of 4. Okay, and you do know that they have that there's this 360, right? And these match because 75% of 360 is 270. So now what does this say about their genotypes, right? So all 270 plants were heterozygous? No, because 25% in fact were homozygous. These are homo. And all 270 plants were homozygous. This is also not true. Only 25% was. Only 90% were homozygous. Also, only 90 plants were homozygous. Not true. All dwarf plants were homozygous. This is true. And actually, this is really easy. Like I did not have to spend this much time because we do know that all recessive genes, when the recessive genes show in the phenotype, they're homozygous. For them to appear recessive in phenotype, for them to appear in the phenotype, this has to mean that they are homozygous. And keep in mind that the result, this is their phenotype. Phenotype. Keep that in mind. Phenotype is what we see in our eyes as humans, and genotypes is what's inside their genes, what's inside their body that we cannot see. So what are the possible phenotypes of children born to a mother with blood group AB? So we know her genotype now. Okay, we automatically know her genotype. And a father with blood group A. So we know only his phenotype. Right, so now we're going to be doing our Punnett square. And keep in mind, this is how we do it. We do IA, IB. Here we have IA. But in this case, we don't know this one. So it can be I for O. So now we know that this cannot be O because no matter what, there's gonna be IA or IB. So cannot be O. So we have IA, IB, IA, IA, let's see, whatever. I and I. So it can be A, it can be B, it can be AB as well. This is like a bit, it's supposed to be B here, A here, but so this is it. Our answer is C. Okay, so I'm going to begin going just a bit faster. So now we have which levels of so we're moving on to topic 4, which levels of ecological complexity involves biotic factors, but not abiotic. So keep in mind, biotic is alive, abiotic is not alive. Okay, so this is where you have to really just know your definitions. Okay, keep that in mind. So community definitely has oh, so these only not abiotic factors. Okay, so this is how you have to literally just read your question really well. So the community is basically just a group of populations. So hence it is technically correct. Ecosystem, definitely not. This involves many, many, many abiotic factors. And 3, population. Yeah, population is just like the same species, basically. Same species. So this is, in fact, D. Our answer here is D. This is our answer. Okay, so yeah. And then how can a chi-square test be used in ecological research to test the effect of an abiotic factor on the plant species? No, this is not true. To test whether 2 species tend to live together, this is true. To test whether one population of a plant is taller than another, this does not test this. It tests how they live together by the measurements and calculations, not the tallness. This has nothing to do with it, so no. This is false. To test whether one species is more tolerant to heavy metals than the other. This is also not true. This has nothing to do with heavy metals. No, it just tests whether how they live together because it just does its basic measurements, like its calculations. We're not going to get into that because this will take a lot of time, but it is B. That's our answer. 18. Under certain conditions, living organisms on Earth produce and release methane. What favors the production of methane? So we do know that it is anaerobic conditions directly off the top of our heads, but this does not, fires does not um, I really favor the production of methods and methane. I want to actually read the question. Under certain conditions, living organisms on Earth produce and release methane. What favors the production of methods? Methane. Okay, so I was just seeing if they're hinting at something specific. Highlight intensities false, anaerobic conditions true, and dry no. So it's anaerobic conditions. And yeah, that's our answer basically, 18. This is a very straightforward question. No analysis required, really. Okay, so now we have question 19. The oceans absorb much of the carbon dioxide in the atmosphere. The combustion of fossil fuels have increased carbon dioxide ocean concentrations. What adverse effect does this have on marine life? Okay, so we're talking about fossil dark carbon dioxide in the ocean, the concentration. So A, heterotrophs consume more phytoplankton. That does not have an adverse effect on marine life. This can be true, but this does not actually affect the marine life in a bad way. Phytoplankton have increased rates of photosynthesis. No, this is not our answer. Coral deposits less calcium carbonate to form skeletons, and also it causes the breakdown of skeletons as well. This is true. An increased pH reduces enzyme activity in marine organisms. No, but this does cause acidity. Oh no, sorry. Because it does reduce enzyme activity, but it's not because increased pH. Because what happens, it does become more acidic, but increased pH does not cause acidity. Okay, so the answer is in fact C. 420, what best explains the formation of different pentadactyl limbs? Okay, so basically pentadactyl limbs in this case is actually when they all have the same feature, but they use it differently. So in this case, it is adaptive radiation. It is not interbreeding, no, because it does not cause different pentadactyl limbs. On the contrary, it causes the same because they're just their offsprings. C, selective breeding. So selective breeding, no, not really. And convergence, no. Our answer is A. For 21, what would restrict evolution by natural selection if species only reproduced by cloning? So basically, what would restrict evolution and natural selection? So natural selection is basically survival of the fittest, reproduced by cloning. So too few offsprings would be produced, or this is not necessary, no. Mutations could not occur, also not necessary. The offspring would show a lack of variation, which is true because it's literally cloning. And our last option is the offspring would be the same sex as the parent. Yeah, but this would not restrict evolution by natural selection because this would restrict evolution definitely, but just not by natural selection. So you have to really be careful with the wording of the question. Okay, so then an animal has the following characteristics: bilateral symmetry, mouth but no anus, ribbon shape. What phylum does this animal belong to? So in this case, it's not a mollusca, not an Idaria. Annelida platyhelmintha. It is basically in this case a platyhelmintha. Okay, so guys, this is it for the video. There will probably be a part 2. But the time is almost up. So I'm going to continue another video just to explain more thoroughly. Thank you guys so much for watching. Bye bye.